One's up. Yep. Yeah. This one, I don't know which one you're trying.
It's Transavia, which is part of Air France, I guess.
Maybe they're fine.
I will check one.
Yeah.
That's why. We are going to start. Excuse me.
Yeah.
Yes, hi.
Can we record the lecture? Do you mind?
No, sure.
Yeah. So please, I think we can start. I think most of the people are already here. So I just come here to introduce you. So today, this morning, we'll have Rick Khan from the University of Birmingham giving the first lecture. So please follow me. You can start. Thank you very much.
Okay, great. Hello, everyone. Sorry, I'm not with you in person. Yeah, I guess the idea here is probably to have a lecture about an hour and then extended discussion. So these are the tutorial objectives. This is what I'm hoping to achieve in the next hour or so. To highlight some of the differences between academic research and industrial R&D. So for some of you, it may be the first time you've had a slightly more industrial lecture. We're going to look at some different types of companies, the different roles for scientists within those companies, and why those companies need you, PhD scientists, PhD level scientists. We're then going to look at sort of classical steps via which an early idea can eventually become a product. And the speed at which that happens very much depends on the application area. And then I'm going to talk about some personal things where I've been involved in small to medium enterprises, startups that have been trying to develop products made from nanoporous silicon. So to achieve those objectives of the seminar, this is how I've structured it. As with all areas of science, there is a language, and some of the words used in the business community may be new to you. So I thought it might be useful to just talk through some of those. And then we're going to look at how product development is different from academic research. From the point of view of career options, I think it's useful for us to think about where our skills lie as individuals. Virtually everyone, I think, is creative, but a smaller fraction of people perhaps are real innovators, and another different group of people are entrepreneurs. And we'll look at some famous people and try and make the distinction. Then we're going to look at different types of companies, big companies, small companies, and how they employ scientists in a number of different roles. we'll look at the product development process, and then we're going to focus in on medical translation, but look a little bit broader as well at industrial translation. And then, as I mentioned, we'll finish by looking at what companies in the past and in the present have tried to develop nanoporous silicon products, what sort of application areas they are focused on, the challenges and the progress. So let's start with some of the language. So I'm sure most of you have heard of things like a business model, which is basically how a company plans to make profits and succeed. And that's based around a business plan. There are different types of products. So, for example, there are incremental products where you're just trying to improve the performance of an existing product. Or there are what's called disruptive products. And these disruptive products tend to be a lot more novel, higher risk, potentially much higher reward. One thing that's very important in companies, even more so than at university, is intellectual property. And companies spend a lot of money trying to protect their ideas, protect their product development progress, and so on. And one of the issues for new companies is whether they in fact have what's called freedom to operate. Freedom to operate means that they're actually legally able to develop a product in a particular country. And you might find that some startup companies have freedom to operate in one country, but not another, because of the intellectual property position. There's something called SWOT analysis, which is often done at the start of a company's development, and is always done when they're considering making a new product. And there you look at the strengths, the weaknesses, the opportunities, and the threats. And you make your decision whether it's a good idea or not to progress with a product based on that type of analysis. So in universities, when we're making nanoporous materials, we make what's called research grade material. And that's in its own right extremely useful. But for products, you often have to make what's called GMP material, which stands for Good Manufacturing Practice. And that material is behind that material, which may on the surface look very much like research grade material. There's a lot more paperwork behind it, and the material is made under much stricter conditions, and we'll talk about that. Nanomaterials are often expensive. And so from a product point of view, it's very important if they have what's called a USP, a unique selling point. from a business perspective, that is. What do I mean by a unique selling point? Literally, it's something which makes that particular nanostructure unique, in the sense that it has a function which is difficult to achieve with other materials. That's quite rare. There are so many materials out there that unique selling points are quite rare. Okay, let's just look at a couple of other MHRA, EMA, FDA. These are acronyms for regulatory bodies who have a lot of power and control over whether you will be able to have a medical product or not. And we'll talk about clinical translation in a bit more detail. Staying on the theme of medical, using nanoporous materials for medical applications, we'll see that to clinically translate, you have to move from the laboratory to patients. Before you can get anywhere near a patient, you have to show that your material that might be a biomaterial, useful medically, you have to show it's safe, and you have to generate what's called a formal toxicology package. And this is a very expensive process, which involves, again, a lot of testing and a lot of paperwork. Okay, let's move on. So that was just a brief introduction to some of the words, the language regarding industrial work. A small number of us think that nanoporous silicon is quite useful from a teaching perspective, because of the diverse nature of nanoscience. We need these educational tools to cover a variety of topics, but also where a nanomaterial has made some progress industrially, you can introduce the differences between academia and industry. And that's what I'm trying to achieve here. So what is entrepreneurship and what makes a good entrepreneur? And are you potentially a good entrepreneur? Is this something that could really be the focus of your future career? It's often about new venture creation, but not always. And good entrepreneurs are really people who are excellent at identifying opportunities, evaluating just how good they are, and exploiting them. That's the key to it. It's and often evaluating is even more important than identifying, because all of us have lots of ideas, and what we need are people who are able to sort of filter a large number of ideas and find the strongest ideas, the ones most likely to succeed. Because most small companies do not succeed. Most small companies in the business world fail, the majority of them. So the evaluation is very important at the beginning, to choose the right idea. So I've got some examples of people here, and some are probably more famous than others, more recognizable than others. And some are not entrepreneurs and had no intention of being one and didn't want to be one. Some are innovators and some are creative. And it's not always immediately obvious what category people fall into. So you might think that entrepreneurs are businessmen and therefore, they normally wear a suit and tie. And indeed, Narayana Murthy on the left, he is a very well-known entrepreneur. But then you have people like Jamie Oliver, who is a chef in the UK, who is a well-known entrepreneur. You have extremely well-known, very creative scientists like Einstein, who is not an entrepreneur. You have people like Charlie Chaplin, who you might think was an innovator, which he was, but he was also an entrepreneur. And Elvis Presley was an innovator, but I would not say he was an entrepreneur. So three categories of person, and which category would you place yourself in is something to think about with regards career choice. So if we look at some of the entrepreneurs and their views, views always vary. But for example, John Caldwell has stated he thinks there are 6 skills you need. And a lot of entrepreneurs talk about hard work and ambition rather than creativity, for example. So this has relevance to if you decide to not stay in academia, but if you decide to work for a company or even become an entrepreneur and set up your own company. There are big companies, small companies, and the key thing is that scientific technology companies, they have a research arm and they have a development arm. So it's a case of choosing where you think your skills and your interests lie. So in a company, the research is all about what's called a product pipeline. So a company has an existing product, but it wants products to come in the future. So it's got a continuous pipeline of products. So the people who are good at research will be coming up with new concepts for that pipeline. The research scientists in the company will be focused on understanding the products. And by having better understanding, they can optimize those products better. They will be heavily involved with IP. So IP is intellectual property, and part of their remit will be to generate intellectual property patents for the company. Now, other scientists within the company are more interested in the products rather than the concepts, and they have more interest in making something that's really going to be useful. And to do that, they look at things like reliability, the volume of product they can make, the cost of the product, and they're more involved with testing, the trials, finding good partners, and so on. Some scientists are good at products, but they're also good at communicating. And they understand the technology and they're able to explain it to customers. So they might then be in sales. And their job is to look at the market, to make sure the focus is right, things like that. So those are some of your options in the future. Once you've got your PhD and you've decided you want to, if you decide you want to spend some time in industrial science, those are some of your options. So this is the standard product development process. You generate an idea, or ideas, ideally, and then you screen them. You bring in people who are very good at evaluating ideas and how strong those ideas are in terms of generating a product. And then that leads to the concept of a product. And then you have to do a lot more analysis. And if it's still looking okay from that analysis, like a SWOT analysis, like a prediction of what revenues you might get from such a product, then you move into what's called product development. And you might make a prototype and you might test that first product in the market. And then you're able to set up a business model. And it's only then that you really start to get serious about manufacturing the nanoporous material on a big scale. So those are the classic sort of steps, and there's some variation depending on what market sector you're in, but that's a sort of classical sequence to developing a product. So let's have a look at some porous silicon companies, past and present. Before we do that, let's have a quick look at some main functions of porous materials and how that links to applications. Can everyone still hear me okay?
Yes.
Yeah, okay, good. So porous materials, be they meso, micro, nanoporous, one of the good functions is filtration. Another is adsorption of things. A third is storage of things. A fourth is controlled release. And they're sometimes used as a barrier or a support. So those are some of the broad functions which can lead a nanoporous material to be considered by a number of different industries. So these are some of the industrial sectors that porous materials in general have been used in, currently used in. And what's key here are whether the porous material has a unique selling point, the amount of porous material that can be made, because some of the applications do not require huge quantities, some of them require huge quantities, and the cost. So often metallic porous materials can be quite expensive. And organic porous materials, for example, can be the least expensive. But they have very different mechanical properties, and there are some applications where porous organics just fail mechanically, could not do the job, whereas ceramic or metallic structures could. So it's a combination of whether they're used a lot is a combination of the properties combined with scale up, combined with cost. So if we look at the abundance and the cost, I'm going to focus on silicon. So silicon is on here. We see that clearly carbon is extremely abundant and extremely inexpensive. But when we compare with metals, silicon is very abundant. We see that iron is an example of a metal that is very abundant, very inexpensive. And what's also interesting is iron is one of the few biodegradable metals. We'll come on to biodegradability in a minute, but you can see that clearly some of the metals like platinum, gold, palladium are expensive, not abundant. They're therefore very expensive, and they're used in situations where, for example, nanoporous gold will really need to have a unique selling point to be used in a product. If we look at the industrial uses of silica and compare them to silicon, silica is very abundant, a natural material. It's available in biogenic form, and is therefore in many more industries than silicon. And so when we compare nanoporous silicon with nanoporous silica, often nanoporous silicon has to have a unique selling point to compete with silica. If it doesn't, then why not just use silica rather than silicon? We're going to be looking at some of the areas where nanoporous silicon has been developed for products, the pink areas. And what distinguishes them is a lot of them have regulatory bodies who control what's going on. So if we look at large companies with porous silicon R&D, how can we find out which large companies work on a particular nanoporous material? A good way to start is to do a search on intellectual property. So a lot of these companies won't necessarily have nanoporous products, but they'll be exploring whether they might. And if they think there's some potential, they will patent their research. And that's a way of finding out whether large companies work on the material you're researching, for example. You'll notice that virtually all of these are electronic companies, which makes sense considering it's silicon. If we look at nanoporous silicon startups, new companies, small companies, they are focused in a number of different areas. The red ones are medical. The brown ones to do with lithium batteries. There are sensor companies. There's one company that offers filtration, and so on, a variety. That was past and present. If we look today, these are the companies that are still active. And as I mentioned, this is a smaller table because many companies fail and disappear. after a number of years. So let's have a look at a couple of case studies. The first is medical device translation. We're going to look at using nanopore silicon in something called brachytherapy. And I was involved with this with a UK company, which did the early research called Symedica, a company in Singapore that did the first clinical trial. And currently this product is now with an Australian company called Oncosil Medical. Then we're going to look at a very different type of company, not a medical device company, but a company looking at consumer care products with a different business model. As I mentioned, there are a number of different types of companies. Some companies develop equipment to make nanoporous materials. Some companies, their business model is straight licensing. They will primarily do early stage research, file patents, and then try to license those patents. And there are other companies that sell material or distribute material. So with biomedical testing, and I think you'll learn a lot more about this area from Professor Nico Vulka, who's talking later. You have what's called preclinical testing, which is everything that goes on before you test your product on a patient. Then you move on to more formal testing. And what you have to decide is your class of medical device. So there are 3 broad classes. And depending on what class your device is, that will determine how much testing you have to do. The longer your device is in the patient, and the more contact it has with the patient, the more likely it is class 3 rather than class 1, and the more testing you have to do. To put a nanoporous material in a human, it has to be what's called GMP material. It can't be research grade material. And there's a lot more paperwork involved. So then we have clinical trials and the regulatory process. Excuse me. So once you've classified your device, you then know what testing you have to do. And clinical trials typically have 3 phases: a safety phase, a phase 2 where you determine the best dose or the best protocol for your product, your medical product. And then a pivotal phase 3 where you really demonstrate the risk-reward ratio for your device, the fact that that it really works, and it works well enough to warrant the risk of implanting your device. And then you get marketing approval, and you can finally use it on patients. So brachytherapy, which is the first one we'll look at, this is highly targeted radiotherapy. A conventional brachytherapy seed is radioactive material in a titanium box. So it's sealed in biocompatible metal, and the surgeon implants it into the tumor, and you only irradiate the tumor. So very targeted radiotherapy. And this was our first medical product using nanopore silicon. And it involved making very heavily doped silicon alloy with phosphorus, micronizing that, stain etching it, then converting the phosphorus into a medical isotope, P32, which is a beta emitter. And the stain etching process had a number of key functions here. We had to have a sealed source. So we needed the radioisotope to be locked into the particle, embedded in the particle, not leak from the particle. The particles were slowly degradable, but biodegradability is not a key attribute for this particular medical device. And this is the progress that was made. The very early stage industrial research was done in 2001, 2002. The first patent was filed in 2002. The formal toxicology took a year. The first safety trial, because it was a medical device that was urgently needed, we were able to go straight into phase 2A and not have phase 1. But you can see, for example, that from 2009 to 2012, there was no technical progress made whatsoever. It was all about trying to raise money for the very expensive trials. And now, today, the product is approved in Europe and a number of other jurisdictions. But my main point in showing this is just how long it's taken. It's taken roughly 20 years to get a product that's approved and can be used. So medical translation is not a quick process. It's a very satisfying area to do research and product development in, but it's a slow process. Let's look at a second type of company that I was involved with. Very different business model and different market sectors, consumer care, food. The business model here was that you pair up with big industry leading companies who have particular problems, and they'll be using a number of small startups to take most of the risk and try and solve their problems. And if you do very well with your technology, then they will financially support your research and your product development. And then they will license your patents, and you as a small company do not have to manufacture, scale up, worry about any of these things. They will do all of that. And your revenue is from your intellectual property. So this was a very different. With Simedica, it was a case of trying to raise the money to internally develop, go through the clinical trials. Here it's about pairing up and letting a big company take over. So these were some of the industrial projects we had. We worked with a very big toothpaste manufacturer. We worked with a big chewing gum manufacturer. We worked with a big chocolate manufacturer and a cosmetic company. And very different industries. These companies will often give you training. So for example, for food, we were trained how to set up a taste panel and assess the taste of nanoporous silicon, the mouth feel, the skin feel, and so on. The lessons learned from these, for startup companies, it's really important to focus, and it's really easy to not focus and to try and do too many things. Some of the big issues with nano products are the cost, how much you can make, how reproducible it is, and how stable it is. And in the case of semiconductors and nanoporous silicon, some of the issues were the fact that if you're anodizing the silicon, your electrolyte is gradually changing. If you're producing the nanoporous silicon by chemical reduction, this is a very exothermic reaction, and that creates problems with scale up. With wet processing, you've always got to dry it at some stage, and that needs a lot of attention. Yeah, these are some of the issues. Okay, so I've left a fair bit of time for discussion. Some concluding comments. So there are quite a few new nanoporous companies in various stages of development. Most of the activity is in the medical and energy sectors. By far, the most activity for nanoporous silicon is with lithium batteries. And there are a lot of companies interested in and looking at and spending a lot of money on bringing the cost of porous silicon down. And that's very good from the perspective of, I think, nanoporous technology. In my experience, clinical translation can be very slow. Clearly, some clinical products can move a lot faster than the one I was involved with. But even so, it is a slow and it's a very expensive process. Quite rightly, there's a lot of interest in greener and greener synthesis of nanomaterials. And I think silicon and silicon carbide are well positioned for this because of the availability of biogenic silica nanostructures. When we first tried to develop a porous silicon medical product, exploiting its biodegradability, there were a lot less biodegradable materials. Now we have biodegradable metals, and some porous silicas are also biodegradable. So there's more competition for biodegradable nanoporous silicon. So the unique selling point is not biodegradability, it's something else. For silicon, it might be ultra-high drug payloads. If we pay a lot of attention on synthesis and drying in particular, we can get very high surface areas, very high pore volumes, and that will translate to very high payloads. Yeah. So I've just chosen 5 articles that I think will give you a flavor of some of the different issues you need to consider for a nanoporous product compared to nanoporous material research. And yes, welcome to now have a discussion.
Okay, thank you very much. So the presentation is now for this session. Please do not hesitate. We can listen to you. Just speak loudly. Can, if you want, turn to the other side just to speak. Please.
Sorry. There's a bit of an echo. I couldn't hear that very clearly.
Okay, we will start now. Can you listen to me better now? No.
Yes.
Okay, so please, do you have questions? Yes, Albert. Can you come here? Maybe it's easier.
Hi, good morning. Can you hear?
Hello?
Can you hear me?
Yes, that's clearer.
Okay. I wanted to ask you about the brachycell. I have a question regarding the etching of the phosphorus. I did not understand very well what was the point of having those phosphorus particles.
Okay, yes. So P32 is a medical isotope. It's a beta emitter, and it has a short half-life, couple of weeks. What we're trying to do is have a microparticle suspension that acts as, from a registration point of view, as a sealed source. So what we want, the ideal particle, is a microparticle 30 microns diameter, where the isotope is within the silicon particle. It's not on the surface of the silicon particle, because then when that sees body fluids, that P32 will leak away from the tumor and go throughout the body. And that's the last thing we want, because we only want to irradiate the cancerous cells in the tumor. So the function of the stain etching, we discovered that when you stain etch phosphorus doped silicon, you selectively remove phosphorus. The second function of the stain etching is, if you just stain etching, you can literally just porosify a very thin layer on the top of the particle. You don't porosify the whole particle. And that modifies the tissue response. So a microparticle with a nanoporous coating, the first time that was put in an animal model, it was found the particles were quite sticky to soft tissue, which again is a desired property, because then the particles basically stay where the surgeon puts them. They don't start to migrate out of the tumor. So we want a very slowly dissolving particle. We don't want fast biodegradability, because that would release all the phosphorus. And we had to show the regulator that there was minimal leakage from the tumor site, which was achieved by stain etching. So does that help?
Yes, yes. Thank you very much.
Okay.
Thank you.
Other questions? Please go close to the microphone, please. The microphone is needed.
Hi, Professor Kehab.
Hi.
Hi, my name is Chechen. Thank you for your very detailed and comprehensive presentation. But there is a problem about the porosilicons, because porosilicon mainly fabricated by these top-down ways, not the bottom-up ways. So mainly because of the based on the electro etching or the photolithography, these ways. So how could we compromise, or how do we compromise, or how could we guarantee the qualities of each nanoparticles? And because of the etching processes, these orientations of these silicons are different. How could we promise that if we wanted to make it into industrial things like this one?
Okay, thank you. So at the moment, there are a number of ways to make borosilicon. Some are top down, some are bottom up. The top down techniques are, like anodization, are particularly good for chip-based products, where you're trying to get a product with a unique selling point. And if it's part of a silicon wafer, a chip,
yes,
then clearly that's quite attractive, because you can have a number of chips per wafer. You're starting with a very substrate that is very pure, very expensive.
yes,
But we know from electronics that that's not a, that doesn't stop you making products. So I think for chip-based products, top down is good. The problem is where you don't have chip-based products, where you're making a simple powder, be it microparticles that are nanostructured or nanoparticles. And then, as you say, ideally you perhaps would like to be bottom up. So some of the lithium battery companies are looking at this. There are some bottom-up techniques. The magnesiothermic reduction technique is interesting because you start with biogenic silica, which is already a nanostructure and relatively inexpensive. But there are issues with that technique as well. So yeah, it's work in progress, I think.
okay.
For very high volume products, it's challenging, even with some of the new techniques coming forward. So I think initially there will be nanoporous silicon products that are sort of medium volume, quite high value products that will emerge. But yeah, so sorry, go ahead.
Thank you, Professor.
Other questions?
Hello, Professor. Thank you for your talk. Can you give me a bit insight about the silica biocompatibility? So I assume it needs to be biocompatible, but for instance, I'm reading about these nanopore smart pearls. So I assume it's silicon nanoparticles that for dermal applications. Are there any issues regarding to these particles on the skin or penetrating the derm or something like that, for instance?
Yes. Most materials have some biocompatibility issues. Silicon is no exception. So, biocompatibility, toxicology, it's all about dose. So we'll often say a material is very biocompatible if the body can tolerate a high dose, but there will be a dose where it's toxic. The biocompatibility is pretty good for nanoporous silicon, primarily because the degradation product is orthosilicic acid, which is the natural form of silicon in the body. I think you'll hear more about this from the other speakers. But, even orthosilicic acid, if the level is too high, can cause problems. But that's the natural form of silicon in the human body. So provided your structure degrades into that is a big toxicology advantage. Orthosilicic acid is also a weak acid. So that means that the local changes in pH are going to be lower than for some other biodegradable materials. And that's probably why the toxicology looks quite good in the eye. The human eye is an example of a site where you do not want large changes in pH around a biomaterial. There's quite a lot of biocompatibility data now available on nanoporous silicon. And I think the other speakers will probably give you more details about that than I have.
Sure, thank you very much.
Any questions?
Hi, good morning. I'm working with graphene materials, and I would like to know how would you compare silicon and graphene in terms of biomedical applications, which is your work is based, but nowadays graphene is also very popular. Do you think that they will be comparable in their performance?
Interesting question. Graphene has some spectacular properties. I know that some of the people interested in biomedical applications of graphene have been trying to explore whether graphene could become, some form of graphene could become degradable. Am I right in saying graphene is not biodegradable? Anyway, that's an interesting area because if you have a biomaterial that's not degradable, the general trend is that more and more biomaterials are being developed that are degradable. So one interesting area for you, I think, would be to look at whether if it's not degradable, then I think that excludes a number of medical applications, but allows some. And are you looking at
Or external, probably not inside the body, but outside the body.
Outside the body. Okay. Yes. Well, yeah. I mean, outside the body, then the chemical inertness can be a big advantage. Yeah, I think it will you know, its biomedical properties will direct you to particular medical uses. Yeah, we'll fact that.
Yeah. Okay.
I'm not sure. I don't know much about how graphene has progressed through. Are there many clinical trials going on with graphene? You probably know more about that than me.
Yeah, I also don't know because I just started to work with graphene materials.
Okay. Yeah.
Okay, thank you.
Some more questions to Professor Lee? Yes, please.
Good morning. Thank
Good
you.
morning.
I like very much your separation of the question about where you are, more researcher or development. I would like to listen to your suggestion for us concerning.
Sorry, I can't. Could you speak a little bit closer to the microphone? I can't.
Sorry, I would like to listen your suggestion for us how we can improve our knowledge in the commercial part, let's say, for development, our knowledge, our production, our materials for commercial, because we are here in university and we don't have direct connection for companies. How we can know about this?
Yes. I think step one is to see whether there are any companies working on your nanomaterial currently. And one way to do that is to use the United States patent database, the USPTO, and it's freely available, and you can search using simple key terms. You put in your material and see whether there are companies, small or big companies, working on it. And if there are, then it's useful to just contact them and have a discussion. Was that the type of thing you meant, or did you mean more sort of formal training?
Thank you.
Okay.
Any other questions? No? If not, we thank again Professor Lee. Thank you very much, Professor Lee. Can you hear me?
Okay, pleasure.
Thank you very much. It's a pity that you could not join us, but it was great to listen to your talk. Thank you very much.
Okay, bye everyone. Thank you.
Bye-bye. Thank you. Okay, so we will have a break. The coffee break is not ready yet, but it will be in, I think, in 10 minutes. And then we'll restart at 11 o'clock, okay? So if you need, just for those that weren't here, the toilets are upstairs. You have to go by this door. You go out of the library because the rooms are inside of the library, and on the right side you will find the toilet, okay? You need. And the coffee break will be served here in this little room at the entrance, okay? And you can go outside if you want, okay?